everyone. This is Haley Rutger again here from Mote's communications team. If you can't hear me very well now, we're going to post a clearer version later from our local recording. But again, this is our web forum on Florida red tide caused by Karenia brevis algae. If we don't get to your question today, you can email it to info@mote.org. And again, that's info@mote.org, and we will do our best to get a response for you in a timely fashion. So I'm going to go ahead and introduce the rest of our group here. We have some wonderful scientists. I'm going to start with the person directly to my right and have him introduce himself.
Hi, I'm Dr. Vincent Lovko. I'm a staff scientist at Mote Marine Laboratory, and I'm the program manager for phytoplankton ecology, working on a variety of phytoplankton in the waters of the Gulf of Mexico with a focus on Karenia brevis, our Florida red tide.
When we're talking phytoplankton, we mean microscopic algae?
Yes, we mean single-celled microscopic algae.
My name is Dr. Tracy Finara. I am the program manager of environmental health here at Mote Marine Laboratory. My expertise is in stormwater treatment design, low-impact design development, and here I work on a number of citizen science projects with red tide, the common name for the species Karenia brevis.
Thanks, Tracy.
Hi, I'm Rich Pierce, Dr. Rich Pierce, senior scientist in ecotoxicology. Ecotoxicology is the study of toxic chemicals and how they affect marine life. Red tide is an excellent example because it affects not only individual organisms around it, but in fact the whole ecosystem.
Hi, I'm Gretchen Lovewell. I'm the Stranding Investigations Program Manager here at Mote Marine Laboratory, and our program responds to calls of sick, injured, dead, and distressed marine mammals and sea turtles, and when they're dead, we try to figure out what happened and get that cause of death and learn from them.
All right, thank you everyone. I'm going to go ahead and start bringing up the questions we've received through registration to this chat and through social media. The community has sent us a wonderful array of questions. You guys are really thinking about this, and we appreciate that. I'm going to start by addressing one of the top questions we've gotten to Dr. Vince Lovko here. What's the difference between the Florida red tide algae, Karenia brevis, and the blue-green algae that we're also hearing about right now?
Thank you. That's an excellent question. So they are two completely different groups of organisms. So our blue-green algae that we see in Lake Okeechobee and some of the rivers resulting from the outflows from Okeechobee are a group of photosynthetic bacteria. These can form blooms under the right conditions, typically high light, high nutrients, high temperature. And these blooms can be very complex, composed of multiple species, some of which are toxic, some of which are not. Some of the toxic species are toxic sometimes and not always toxic. It really depends on the conditions. This is very distinct from our Florida red tide, our Karenia brevis, which is another group of plankton, of phytoplankton, dinoflagellate. Our blooms here are typically monotypic, meaning they are composed of one species. And this species, of course, is Karenia brevis, and it produces a toxin called brevetoxin. And this toxin can, of course, it's a neurotoxin and can cause effects in humans through respiratory irritation, and also can kill fish and other marine organisms.
Right, and people have been asking if there is any relationship between Karenia brevis, red tides, and the blue-green algae that we're hearing about. Do you know if there's a relationship?
Well, that's a difficult question. So, typically these organisms would be in very different environments. Of course, the cyanobacteria are in fresh water. We do have marine cyanobacteria as well, and there can be a relationship there. There's a species called Trichodesmium that typically forms blooms offshore. It can survive in nutrient-poor waters because it actually is able to pull nitrogen out of the air, one of the very important nutrients, nitrogen. And that can form extensive blooms. These blooms can eventually sink to the bottom and die, and it's thought that that release of nutrients from these dying blooms could be a trigger to the initiation of Karenia brevis blooms. There's also another marine marine cyanobacteria called Synechococcus, and some laboratory studies have shown that Karenia brevis can feed on Synechococcus, and if Synechococcus is present in high enough volumes or high enough concentrations, then that could potentially provide enough nutrients to Karenia to sustain a bloom.
Now, let me stop you real quick and say both of these live in the ocean, right?
Yeah, both of these are marine cyanobacteria.
Yeah.
Now, as far as an interaction between freshwater cyanobacteria and Karenia brevis, well, normally that doesn't happen. Is it feasible that large amounts of cyanobacteria coming, for example, through the Flussahatchee, if they actually make it to the coast, could that the degradation of them fuel phytoplankton in general and Corynea brevis specifically. Well, sure, but that would very much be a hypothesis, and we have not demonstrated that connection.
A hypothesis, something that we would need to test through further data
Right.
collection.
So, in science, we make observations, and those observations might lead to questions, might lead to the idea of, can this happen? And that's when we form a hypothesis. We design studies to test that hypothesis.
So right now, I want to address a question to Dr. Rich Pierce over here, if you can pass the mic. Right now, we're looking at some really concentrated cell counts of the red tide algae Corynea brevis along our coast. We know that blooms form offshore and they can come to shore, and that's definitely happened here, and the cell counts have been pretty high. Do you know if the current red tide bloom is the worst or one of the worst that we've seen?
Well, I can honestly say that it is a very severe bad red tide at this time. As far as my recollection, it is not the worst that we've had. In fact, as recent as 2004, 5, and 6, we had an 18-month boom and a tremendous amount of fish, marine mammals, turtles were dead, and it caused a lot of problems. This one has been going on and off for about 9 months, so we're hoping that it won't break a record, but that's something we're still waiting to find out. So this is a very severe red tide. It's not the worst we've had. It seems like every 5-7 years or so we get a bout of pretty bad red tides, and then we're able to have a reprieve for a while.
Yeah. So, and when you say, when we try to decide what's the worst, it's not simple, right? It's, you can talk in terms of how long they last. You can talk in terms of how concentrated they are. In this case, Rich was talking about how long, but there's a lot of ways we could describe a red tide, right?
Well, that's true. The concentration is not something that's static. It goes up and down. It moves. A very intensive patch of red tide will move up and down the coast. And so you're at one beach one day, the next day there might be very little red tide there and a whole lot of it just about 10 miles up the coast. So just looking at a cell count at one place doesn't give you a good idea. Whenever we have extremely high concentrations staying in a place for weeks and months, it causes a lot of problem. And we've had that happen with this bloom.
So I have a question that I want to field to either Vince or Rich, or maybe both of you in turn. People are asking if red tides have grown worse over time. They've been pointing out a publication to us, a 2007 article in the journal Harmful Algae about a long-term increase in Corynea. And I wanted to understand, as scientists, are you able to tell if that's true, if there's a trend of worsening?
Well, it's a difficult question to answer, or it's a difficult topic to address. Trying to compare modern blooms, more recent blooms to historic blooms, partly because sampling has changed. Both our sampling effort, the amount of times we go out to sample, the number of samples we collect, the technology involved in both detecting blooms and monitoring them, and also our ability to go out and sample. I mean, something as simple as a change in the type of boat we use has allowed us to cover much more ground since about 2012. So when you're comparing older data to newer data, that has to be taken into account. It's not clear that study referenced adequately took those factors into account. That study also went through, I think it was 2002 as far as the data collected. Certainly there's been 16 years of data since then, 16 years of monitoring and data collection since then, including several major blooms. So it's certainly something that should be revisited. I don't think we can conclusively say that blooms have gotten worse, but we also certainly can't say they have not.
Right, and I mean, any bloom we say that affects wildlife and people is tough enough, no matter where it fits on the spectrum. I wanted to ask a question that we've been getting really often on our Facebook to Tracy here, to Dr. Tracy Finera. People are asking if water releases from Lake Okeechobee through the Caloosahatchee River have done anything to exacerbate this bloom. Can we know?
So the Lake Okeechobee releases have been going on for a while. When South Florida gets a lot of rain, like they did in May, water is released from Lake Okeechobee when it gets to a certain stage to the St. Lucie and Caloosahatchee River. Along with water and runoff from the Caloosahatchee watershed and those of Lake overflows, we see that those waters may not have had the degradation time needed to biologically or physically remove those nutrients from the water body. And we see that, we know that because the ecological response of the water body in cyanobacteria blooms. Sorry. So we know that water does have high nutrients and comes to shore, but we know that coastal waters are high in nutrients, and we know that because we have a lot of phytoplankton activity and productivity, and that's why we have such good fishing. However, of course, too much of a good thing is always not good. So, so we know that blooms initiate offshore, but if they do come close enough to shore to outcompete other species and uptake those nutrients, it's very possible that they can use those nutrients to sustain duration. But there's difference between they can and they do.
So Tracy, you're saying it's possible for the Karenia brevis red tide, if it moves to shore, to use nutrients that are both natural to the estuary and that humans put into it. But
Yes.
whether a given bloom has done that is not so easy to say?
You're exactly right.
Okay.
Nutrients are nutrients, whether they are man-made or human or natural, and that's exactly what I'm saying. But it's also really important to note that there are many outflows of surface water flows throughout the Gulf Coast and throughout Sarasota County, and a lot of those nutrients that come out of these water bodies are quickly degraded. So the best thing that we can do is really take a look at our stormwater, take a look at our what we're putting on our lawns, take a look at all of those nutrient inputs and try to retrofit the community's low impact development design. Of course, I'm going to say that that's my expertise, but to remove those nutrients at the point source to prevent from cyanobacteria blooms, of course, and perhaps to not feed an existing red tide bloom, but again, we just don't know what kind of effect those surface water nutrients have for any of this.
Yeah, so it's what you're saying is that no matter what, no matter exactly what it might do for a Karenia brevis bloom, excess nutrients aren't going to be good for the environment, right?
Right.
Right. So
Too much of a good thing.
too much of a good thing. So we need more data, I think, but we would also say if we can reduce nutrients, that's a good idea. I wanted to ask Vince, because Dr. Vince Lefko here, what causes red tide to form? We were talking about whether it could be...
I get the hard question.
You get the hard one. Whether we were just talking, Tracy was saying whether it could be exacerbated by coastal factors, but she mentioned that it forms offshore. So what do we know about that?
Sure. Well, our current understanding, and scientific understanding changes when we get more information, but our current understanding is that blooms initiate near the bottom offshore. It could be as much as 10 to 40 or 50 miles offshore, and that gives you an idea of how uncertain we are about exactly where they initiate. And what the initiating trigger is, I referred to trichodesmium earlier, but there's a few other hypotheses as well. But this is generally thought to be outside of the influence of land-derived nutrients, so anything that humans produce or even naturally produce nutrients from the land. So exactly what that trigger is, we don't fully understand. Like I said, there are several hypotheses. I could get into them if you like. I might take up the whole hour.
Might take the whole hour. But okay, so what I've heard is that they can start 10 to 40 miles offshore. Is that right?
Yeah, generally.
Okay.
I mean, it varies depending on which scientist you talk to. But that's the consensus.
Okay, gotcha. So basically, if I'm getting this right, it forms so far offshore that it's distant from the coastal sources so much so that we wouldn't think that they're the reason it starts, but once it gets in.
Right.
Yeah.
So that area right near the shore, has a lot of other phytoplankton in it, and it has a lot of phytoplankton in it because they are taking advantage of nutrients that come from land. Even with Karenia brevis, there's been a lot of nutrient sources that have been identified. There was a paper that involved some Mote scientists as well as other collaborators, 2011, I believe, that identified 12 different potential nutrient sources. And one of the main ones is water flows carrying nutrients from land. These are natural water flows, natural nutrients. Those, as Tracy was talking about, those can be enhanced in a variety of ways, not just through something major like the flows through the Caloosahatchee. And if we increase those nutrients in those coastal systems, then that provides more food for the phytoplankton, the algae that are there. And that could also include Karenia brevis when there's a bloom.
Can I ask, I've heard that Karenia brevis has been around longer than us, something like that, that blooms have been existing since early colonization of Florida. Can maybe Rich give me some comments on that?
Yes, actually, there's evidence in 1500s of the Spanish explorers' logbooks of things that were described to represent red tide event. So we know it's been around a long time. And of course, we have evidence back into the 50s and before that actually one of the worst blooms occurred in 1947, right after the war. The airborne or aerosolized neurotoxins that are produced by the organism were so bad, people were complaining that they thought the government had dumped nerve gas off the coast of Florida. But scientists from University of Miami and from Woods Hole Oceanographic came down, and actually that's when they first discovered what was the cause of it. And at that time, it was called Gymnodinium brevi. So a lot of research has been going on.
Great. Well, I wanted to move on to some of the impacts of the red tide, especially that we're having now. And Gretchen Lovewell is here with us to talk about the wildlife that we've been responding to. It's been an extremely busy couple weeks for you guys, hasn't it?
Yes, it has. Over the last couple weeks, we've picked up 9 dolphins. We've assisted with recovery of at least 3 manatees and I think over 40 sea turtles in just the last 2 weeks.
And as far as a bloom of red tide goes, is this a pretty significant impact for what you usually see?
I mean, compared to historical data, yes, we're at extremely elevated levels. Our sea turtles are running pretty similar to what we saw in 2004, 2005 in our data. For our local area with the dolphins, it's been a little different. We hadn't seen that so much in that 2004, 2005, or 2005, 2006 bloom. That being said, there's been several unusual mortality events that have been attributed to red tide with dolphins, primarily up in the Panhandle. There were 4 in the Panhandle, 1 in the central west coast of Florida. So it's not unprecedented to see the dolphins. We just, I think, got a little lucky in the early 2000s.
Yeah, and from what I heard this week, it sounds like you're working to confirm whether or not red tide is the factor. Like you have to do necropsies and take samples, right?
Right. So for all of the animals we recover, especially if they're in decent enough condition, they're not too decomposed, we do extensive postmortem exams. And talking to our colleagues with FWC, we are confident in saying that that is what's causing the turtle deaths. They've done enough previous work, and we've spot tested some of the animals so far that is definitely, we're 100% okay saying that. For our dolphins, that's our highest differential right now. It'll take us several weeks to get those data or those analysis back. Much like us, I think the folks that are running those tests are just as backlogged because they're getting not only dolphins and turtles, but water samples and things from all over the area. So
yeah, you guys are doing incredible work. You're not a huge team, but you're responding day and night and doing necropsies here, which are animal autopsies. Important work. We had questions on our Facebook page about sea turtle hatchlings, whether they're vulnerable to red tide, and if they are, why can't we maybe hold on to them until the bloom passes? What can you say?
That's a question we get a lot when we're out there responding even, and what we're pretty comfortable in thinking is happening is, those baby sea turtles are actually born with their own food source. It's in their yolk sac. So, and when they hit the water, they go through a swimming frenzy. So what we're thinking is happening is they're getting out there and they're actually using their yolk sac and getting past the bloom and getting to the weed line where they will spend the first part of their life. I think we haven't seen elevated numbers of dead baby hatchlings on the beach. So I think that's a pretty good assumption that's what's happening.
Okay. That's a sign I'm happy to hear. In general, we are, checking into suspecting which deceased animals might have been affected by the Florida red tide. We're not talking right now about deceased animals in our area and, say, blue-green algae. People are asking, though, if blue-green algae could also play a role in some of the fish kills, sea turtles, or even the dolphins. I don't know the answer to that. Is there a way we can know that?
We don't know that, at least from the animals that we focus on, the marine mammals and sea turtles. I actually was trying to look some of that up, and I kind of ran into some dead ends. With the blue-green algae, I think they've shown it to affect freshwater sea turtles in areas, but it's been pretty hard to find, so I just don't think we know well enough yet.
Okay. And we do know, though, from our monitoring that we have some pretty significant Corynea brevis red tide counts here, though.
Yes.
Okay.
Yes, we do.
Gotcha. All right. Well, I wanted to ask a question to Rich, something that's on everybody's mind right now. Can we make it go away? Can we make red tide lessen or go away?
Well, that's a good question, and it's a naturally occurring phenomenon, and so it would be very difficult to go out into the Gulf of Mexico and do something to make it go away. People say, well, can't you just spray it with something and kill it? And
Right.
of course, we could, but we have to be careful because it's part of a natural ecosystem. We got to be careful that we don't mess that system up, because if we get rid of red tide, what else might take its place? We don't know. There are things, however, that we can do near shore in smaller areas, and we've actually been testing a lot. Several years ago with Woods Hole Oceanographic, we did testing of some clay. Clay, it causes a flocculation, and as it filters down through the water, we found that it absorbs the toxins and takes them out of the water, and it also causes the cells to lyse, and in many instances to go to the bottom and die. However, the toxins are still there, and if you're a clam or a worm sitting in the sediment, you're not real happy about all the toxin coming down.
No.
But it worked well around Japan and some of those areas where they have deep fjords and they have fish pens that they're working on. But here in Sarasota Bay, that clay didn't work. We've also been working on some chemical effects. We do know in our aquarium we use ozone to purify the water. And for the Marine Mammal Hospital and Sea Turtle Hospital, when red tide is in the area, we still pull the seawater in, and we put it through a very sophisticated ozone contact purification system. So we looked at that and we said, hey, why can't we do that perhaps out in a limited area in the field? So we tested the system with our 25,000-gallon pool, or mesocosm, as we call it, and we found out the system works very well. We were concerned that we might be producing more nutrients by oxidizing the organic nitrogen and phosphorus to nitrates and phosphates. If we did that, we'd just be eating more fertilizer. We found out that doesn't happen, and so now we're getting ready to do our actual first field test in a canal down in Boca Grande. So we're pretty excited. This is something that would be used in limited space, but it might help restore some of these residential canals, which are dead end, very low flow, and they just become putrefied during a red tide with all the dead fish. There's other things going on, and I might let my colleagues talk about some of the things that they're working on.
Yeah, I was actually going to go to Tracy next, conveniently. So Tracy, can you give us a rundown of broad brush strokes of what Mote's doing in regards to either better understanding or better addressing red tide?
Okay. One thing I want to say as far as Rich's system, it's definitely a contained system.
Yes.
We want to stay away from, if we kill red tide, chances are we're killing everything else with it. That's one thing to really
So it really,
keep in
you're
mind.
saying it does not release ozone.
Right. It
Yes.
does not release ozone
happening
to the
inside.
natural environment.
Yeah.
So on that note, we're doing a few things to first of all, not only educate and inform the public, but also to mitigate effects of red tide. So at this point, it's naturally occurring, and the best thing that we do right now is inform the public of where those effects are so that they can make healthy decisions when going to a beach. So we have the beach conditions reporting system, which was first developed in 2006, and then when I came to Mote, we developed it in 2015. It shows beach parameters, including red tide effects of respiratory irritation in dead fish. These reports are made by our beach sentinels, lifeguards, facilities, and we're so grateful that they are willing to help out by informing the public. We have an app on Android and iTunes, and then we also have the website, visitbeaches.org.
Hey, Tracy, I heard from, Rich and I were on a call yesterday. We heard that the beach conditions reporting system has gotten something like 1 million hits during this bloom. And I think you said there were hundreds of thousands of users.
Yes, so wow, we've gotten over 1 million hits on the beach conditions reporting system, and I think 350 or more than that users since the beginning of the bloom, which was mid-October, something that we haven't really said outright. That it's been around for a long time. Oh, actually, I think Rich did 10 months. And here's the thing with red tide, it's really patchy. So you can be on one end of the beach and experience effects and not on the other end of the beach. You could be at a beach a mile up and experience effects and not at the beach a mile to the south. So to fill in those blanks, Vince and I thought of a citizen science reporting app called CSIC, C-S-I-C, that people can report red tide effects, like respiratory irritation and dead fish. And then it's kind of my, because I want to make it into this whole environmental reporting app with water quality as well, but we'll talk about that another time.
What does it stand for, Tracy? C6?
CSIC stands for Citizen Science Information Collaboration. Really, we wanted to make CSIC work. But in addition to those citizen science reporting apps, which give the public qualitative reports, we have HabScope. It's a NASA-funded project that we are contracted by NOAA and GCUS on. And what it is a citizen science project where we train volunteers to use a cell phone microscope. They take a sample, put it underneath the microscope, upload a 30-second video. The algorithm in the app that they're using calculates Karenia brevis, or red tide, concentration by its shape, size, and swimming pattern. That information then goes to a NOAA respiratory irritation model so that we have real-time respiratory irritation data. So that's pretty exciting. It adds quantitative data to our qualitative reports. In addition, we're trying to use biofiltration or living boxes, we're calling them artificial reefs, whatever you want to call them. They're structures that are colonized by filter feeders in the natural environment that we're hoping will alleviate some of the respiratory irritation experienced during low, medium, and maybe right at the first part of the high cell concentrations. What these filter feeders do is remove Karenia brevis toxin from the water column.
So Tracy, the toxin, the brevetoxin, is what makes us cough, what makes sometimes our eyes hurt. If you have asthma, that's what can trigger your symptoms. That's all the brevetoxin that comes into the air?
You're exactly right, and we really haven't mentioned that much, that what makes Florida red tide so special. There are red tides reported all over the world, but Florida red tide is specific to Karenia brevis, and what makes it so unique is that the toxin does aerosolize, attach onto sea salt particles, and go over a mile inland and cause respiratory irritation in healthy individuals like coughing or sneezing. But for those with asthma or COPD, this can be very serious. That's why getting that information to the public is so important.
Yeah, and we're seeing a picture of right now what looks like our beach conditions reporting. We just took a look, and you can see all the beaches that we were monitoring. It's 30-something right now, right?
Right.
So we have all these tools, these different monitoring tools, and then we're exploring mitigation or control, how to actually address the red tide. We also do monitoring out at sea. Dr. Vince Loveco and his team collected hundreds of samples from this bloom. So there's a lot going on. Now, I wanted to address a question about funding. We had people online asking us who supports our red tide research. First, I'm going to ask Rich and Vince to say who does fund our red tide research? Where do we get most of the support? Well, any of you guys can address that. You can each take your turn, yeah. Go.
All right, well, I'll start. I've been here in red tide funding longer than
Yeah.
But our base funding, the primary source of our funding, is a collaborative red tide research program with the state of Florida Fish and Wildlife Conservation Commission. The state of Florida Fish and Wildlife Research Institute has many very, very excellent scientists that work with us, and we get the state of Florida provides funding for us as a collaboration. Now that is what primarily right now is used for, say, public health, public information, and more environmental health. Now we use that base to, of course, maintain our scientific facilities, some of them, and our staff, but we can leverage that funds now, and as Tracy said, we have funding with NOAA and NASA, and we also have funding from other sources as well, and this allows us to extend beyond just the public health information, and we can start looking at things that we've talked about here, maybe mitigation, even control aspects of the tide.
Great.
Hi, everyone. So my funding comes from NOAA, NASA, GCUS, County, Visit Sarasota County, Collier County, the state, of course, and START has helped with our exhibit and with our BCRS.
As Rich mentioned, a lot of our funding for our based red tide work comes from our collaboration with the state of Florida and FWC, FWRI. And a large portion of that is the monitoring efforts that we do. We execute a lot of the monitoring for the state. And that monitoring data is really important, both in the short term as far as detecting red tide, ideally, in an ideal world, providing a little bit of an early detection system. We monitor year-round, not only when there's a red tide, but also over the long term that builds up a data set, because we're looking at more than just the presence of Carenia brevis and the amount. We collect a lot of other parameters as well, including nutrients and other water quality parameters, toxins, phytoplankton community. And all of those together over the long term allow us to look at those conditions that are associated, with our environment, and then how those relate to the presence of red tide. And the idea is to help us better understand what might initiate a bloom and carry on a bloom, but also over the long term, hopefully help us develop prediction and forecasting tools.
Yeah, that's good. And I want to mention that there is a level of forecasting that FWC and US at University of South Florida do right now based on water movement. They can tell us 3.5 days out where the water might be going, where it might be carrying the red tide. But even better forecasting is everyone's dream, I think.
Yeah, and generally speaking, because harmful algal blooms are a problem worldwide, and among the harmful algal bloom scientific community, that's where things are moving. There's a big push to start to develop these tools for prediction and forecasting, because that's one of those mitigation tools that can really be helpful. Understanding when and where a bloom's going to be is very valuable. You could equate it to weather. That's one of the things that are attempted to do with weather, is to get better at predicting and forecasting. We know when severe weather is going to hit, we can better prepare for it.
Yeah, so that's a really great goal. Before we move on from discussing funding, I wanted to answer a question that a lot of people had asked on our social media, and here people are asking if and why we would get funding from the company Mosaic. They produce fertilizer, which is why people are asking. And none of these guys get any funding from them. I'm not sure any of you even knew there was any.
I didn't even know what Mosaic was.
I did not.
But okay, yeah. But our education studio that we're in got some for its development, and in the past, one of our fish farming research programs got some funding from them, and so did a couple of our events, especially our run that ultimately raises funds for sea turtles. So we just wanted to make sure that if you're listening and you're curious, that you know what that was, but it doesn't relate to these guys. We have to remind everyone, Mote's independent, and our research is only of value if we are independent. So we just want to let you know that. But the next thing, probably the most burning question, is what's going to happen next? What's going to happen with this bloom? Can we say what's going to happen and how long it'll last? I don't know, Vince, can you help me with that one?
I'm holding the mic, so I guess I'll start. That's a great question, and the short answer is we don't know. Again, we need those prediction and forecasting tools that would certainly help us. What we do, frequently what we've seen with red tide blooms, Carenia brevis blooms, is there had seemed to be a season that often starts in like late summer or early fall and lasts through winter. Often it seems with a break around December for some reason. And then, picking back up and lasting into, maybe March or April. There's, of course, variation to that. Again, as Rich had pointed out, or has been pointed out several times, there have been blooms in the past that have gone beyond that season and lasted through an entire year into the following year. Whether or not that's what we're going to see this time, we don't know. We certainly hope not, but we don't know.
Right. In the meantime, where should people look for information? I know one source is Mote's Beach Conditions Reporting System, but where else can they look for what the bloom's doing? Would that be FWC?
There's a variety of sources. We could all say some of them. Of course, there's in addition to the Mote website, there's FWC. NOAA has a forecasting system that's geared towards respiratory irritation. There's, of course, the BCRS. Others chime in.
Did a
Pretty good job. And CSIC.
And the USF, did you mention mentioned the USF.
Right. The modeling that USF does, the 3.5-day model that Haley was talking about, is available on the USF website. Just get that information. Cool.
Yes, we can. You can actually go to mote.org/redtide. You can find links to resources there along with our FAQs that we're working to update based on the latest research information. And I guess I wanted to just finish up with a few practicalities for our viewers. For instance, Tracy, I was going to ask you, even though we still have to answer some questions about how water quality affects red tide at the coast, the point is we still need to improve our water quality. Are there some recommendations you can make for how we can do that?
Yeah, regardless of the influence on red tide, if we are still getting those cyanobacteria blooms, which tells us that there are high nutrient loading going into our water bodies. And the best thing that we can do is take care of that nutrients at the source. So fertilizer mandates, which a lot of cities already have. I know in Lakewood Ranch in Sarasota, you're not allowed to fertilize during a wet season, which is always beneficial. But also low impact development design, which basically takes out that, I know I'm going to use a word here, but directly connected impervious surfaces. Now, it sounds complicated, but really it just means that when water hits your roof, goes down your gutter, goes into your driveway, and then goes into a pipe. And all that water that goes into the pipe is not treated at all. Every drop of water in the state of Florida that falls on the ground either infiltrates into the ground or ends up in our natural water bodies some way through pipes or natural systems. So it's really important that people know that, first of all, so that they can make decisions not to throw their garbage out on the street and make decisions when they're putting things on the lawn or when they're debating whether they should pick up that dog poop or not. So it's always good to limit those nutrients and disconnect those impervious surfaces, allow that water to infiltrate into the ground, recharge that groundwater table, or if you're in a place where you have really high groundwater levels, there are exfiltration trenches, bioretention gardens. You can even retrofit your own house. There's so many different things that we can do right at our homes to minimize the amount of nutrients that we're contributing to this problem.
So, Tracy, I was recently on the Science and Environment Council of Southwest Florida website. They have their watershed tour and tips about how to keep the watershed clean and to help with some of those things. They talk about using porous surfaces and choosing plants that need less fertilizer.
Yes,
so
that's all low impact development.
it's a good resource, right?
Yes,
okay.
Absolutely.
So you can check out the Science and Environment Councils of Southwest Florida website to get some of those resources. And then I was going to ask Gretchen if people find an animal that they think is affected by red tide or affected by really anything. If they find a marine animal that looks like it's in trouble, what should they do?
Well, if you're in our local area, by all means, call our hotline, the 941-988-0212. They can also call the FWC Wildlife Hotline, 888-404-3922. These numbers memorized. The biggest thing that we need when you call us is, what are you seeing? Not just the animals dying, because we need to understand what you are actually seeing. You know, if you see a manatee and it's in brown water and there's dead fish, but it's flat and level in the water, seems responsive, is breathing every 3 to 5 minutes, that's not an animal we're concerned about. It doesn't need our help. You know, manatee mating herds are happening right now. They're sometimes coming close to shore so that female can catch a little bit of a break from the multiple males pursuing her. They're okay. That's natural behavior. So a lot of our time's being spent teasing out what's normal and what's not. But by all means, if people are concerned, we're happy to talk to them, take those calls, try to tease out what's happening. But, you know, I just want people to understand that when we're asking these questions, it's not because we don't believe them that they think something's wrong. We're trying to ask sort of non-leading questions to get a true description of what they're saying. And, you know, better yet, this day and age of smartphones, a video, pictures speak 1,000 words for us. If we can see a video and actually hear and see what that animal is doing, that helps us sort of target our resources and send people when the animals truly need help.
Thank you. That's really, really good advice. Did anybody else have anything to add before we wrap up today?
I wanted to mention our ocean technology program. We have some tremendous engineers that are designing and using some fantastic, sophisticated robotic type of instrumentation, so that instead of having to take a ship, a bunch of people out month or so, we can send these autonomous vehicles that monitor for red tide and send back information. So we have now a brand new programmable hyperspectral seawater scanner, which is a very sophisticated piece of equipment.
AKA the
using
fizz.
right now.
We call that the fizz.
The fizz, right.
Yeah.
So I just wanted to mention our ocean tech. Fantastic tech.
Yeah, really important component of what we do. Yeah. And
should-
go ahead, Tracy.
Sorry. I was just going to say we should probably mention the other kind of things that we're doing to mitigate red tide. I mentioned the lift jacket. I know that Vince is, my other stuff is kind of too preliminary to even mention, but I know that Vince is already doing stuff with macro algae that you can talk about.
Well, Rich had mentioned the difficulties in trying to do something about a red tide. And that's when we have a red tide, especially when it's this severe, people want to know what can we do about it? How can we make it go away? Well, if you think logistically, our current bloom is somewhere around 150 miles long, roughly maybe 10 to 20 miles wide, and I've calculated it out, and that comes to about 1.5. Well, if you look at just the upper meter, which is where we generally think most of the Karenia would be, although it can be throughout the water column, and even at the bottom in pretty deep waters. But in any case, that volume of water is about 1.5 trillion gallons of water. So if you try to think about how you would treat 1.5 trillion gallons, that's an enormous amount of material that you would have to apply to a bloom that size. However, as scientists, it's our obligation to find out what's possible. Practical, might have to take a back seat for a while we figure out what's possible. So one of the things that I've been exploring, my program has looked into, and it's based on the literature, some things that are have already been looked at with other species. So macroalgae, the things we often refer to as seaweeds, seem to produce compounds. We currently think those compounds are something called polyunsaturated fatty acids, that these things are inhibitory and even algicidal, meaning that it kills algae towards a variety of microalgae, but including Karenia brevis. And so we've been exploring that in laboratory studies. Now, whether or if we'll be able to take that to field studies, that's in the future.
That's that's really interesting. We got a very interesting question from one of our guests here today. Does red tide affect drinking water? Who can answer that one for me?
No.
No.
No, however, that being said, we already know that some species of cyanobacteria can be harmful, and like people in around Lake Erie or the city of Toledo have seen, and I've heard that it's happened in other states as well. Those toxins can affect drinking water. Red tide is marine.
Mic up.
Oh, mic up.
There we go.
And now, when it comes to reverse osmosis systems, where we're taking ocean water, those advanced water treatment systems would normally remove that toxin, and we wouldn't have to. So no.
No for the Karenia brevis red tide. Now, I have to say that the term red tide gets applied colloquially to a lot of different kind of algal blooms, so we have to be careful about what algal bloom we're talking about. Is it freshwater? But for Karenia brevis red tide, that's a no. All right. Rich, were you going to add something to that? No? You just got the mic? Well, I have one more question from our guests today, which was, if this red tide bloom has been around as long as we've been reporting it, which was late October to early November 2017 through now, why are some people just noticing fish kills now as opposed to way back then? Why do some people only just start seeing them?
Well, we've seen that before in I think it was 2000. The bloom we had in 2012 that started a bit more at a expected time. I think it was September. We got it to its worst in October, November, and we didn't really see that many fish kills, as I recall, earlier in the bloom. It was later, and I think basically as it has time for that bloom to stick around, producing a lot of toxin, the fish to be more exposed to it. And also, a lot of times these blooms can be very patchy, and the bloom we're currently experiencing was quite patchy. Back in November and December, it was pretty solid. It started to, although it was much smaller than it is now. And then in January and February, it was very dispersed along the coast. In fact, in February, it made it all the way down into parts of the Everglades and even the Keys. Again, it was very patchy. That wasn't like a solid streak of red tide. So as the ensuing months, March, April, May, and the summer came, that bloom got much more widespread and much more solid, much more consistent along the coast. And I think that's probably just that consistency of the bloom and the amount of toxins in the water and the inability of fish to be able to escape it is probably what's caused us to see a lot of these mortalities.
Well, thank you. I want to check in with our studio folks. Do we have any more questions? Standing by. Do we know if the Mississippi River flow influences offshore blooms of red tide? Have we studied that? No?
We haven't studied that. There has been, I'm trying to remember how long ago it was, but there was a hypothesis at one point that the Mississippi flows could actually carry nutrients across the Gulf and push them up onto the shelf. To be honest, I'm not quite sure where that research stands or if anyone's further explored that.
Yeah, to my understanding, there are several hypotheses about bloom formation, and that's one of them that we've heard over the years.
Yeah, I mean, there's many, and you know, most of them tied to a source of nutrients. There's submarine groundwater discharge, where water that seeps into the ground eventually makes its way out through some of these old spring sites, many of which are located offshore, and that water pushing up through the sediment can bring nutrients that can spur Corynea growth. There's the trichodesmium one that I mentioned. There's even a dead fish hypothesis, to where as a bloom starts and it's growing and producing toxin and killing fish, that those fish die, decompose, and then can further feed the bloom. It sounds a little outlandish, and if you think about it, in order to have, as we just identified, in order to have fish kills, the blooms usually has been going on for a while. But then other times when you look at the surface of the water during intense bloom and you see that scattering of fish, you know, in some cases, as far as the eye can see, you can understand where that hypothesis might have come from.
Thank you. How about our studio? Anything else? Okay. So we're asking if any agencies that we know of, set up warning signs about red tide on waterways. I think we've seen some beach signage before. Do we know an answer to who would do that? Do they always do that? That kind of thing? I'm not sure.
I know that Sarasota County beaches put up a notice when red tide's in the area, and sometimes there's a particular flag that goes up to indicate a hazard. We've been asked, should we see about putting up signs warning people? What we do is just tell them what the situation is and are inhaling and feeling irritation. That's not to stay there. We've done a lot of work on the aerosol, and we know that if you use a painter's mask or surgery mask, it will filter out a lot of the airborne neurotoxins.
Right. That's a good advice for a lot of us. But if we had asthma, the Department of Health might tell us to stay away from that beach, right?
Yeah.
Yeah.
The neurotoxins are also bronchoconstrictors.
That makes sense.
We also want to mention foam along the beach. Be careful to keep the dogs out of that. Don't let them lick dead fish that are covered with foam, because scientists at Mote actually discovered the actual process by which these very large neurotoxins get into the
Tracy's
into
pointing
the
at
air
Rich.
by a bubble-mediated transport mechanism, and there's a lot of bubbles. They get stuck in the foam and come on shore. So the foam can be very highly concentrated in these neurotoxins. You want to keep children and your dogs and animals out of it.
That makes good sense. We got one question in that kind of relates to what Vince was saying about fish. There's a hypothesis that fish may contribute to the algal blooms. Do dying fish, dead fish, and other organisms contribute any nutrients that we would think would influence the blooms?
Well, yes, they do. As they decompose, the nutrients are released, and that could very well enhance a bloom.
That makes sense. Any other questions from our studio? So we're getting asked, what can people do? Do we need volunteers? One thing I know from talking to you guys quite well is that our CSIC app, our CSIC Citizen Science app, could use more people reporting on it and alerting us to where they're seeing bloom impacts. Are there any other calls for help that you guys want to put out today? No? Okay. Tracy's got one.
So if we do get funding to further our ABSCO citizen science project, it would be good to have a person at every beach that we have reporting on the BCRS. That would be ideal. Now, these people would need to be able to use a microscope and take samples daily. Beach Conditions Reporting System, just to, in case you're tuning in right now, visit beaches.org, or the Beach Conditions Reporting System on Apple Play or iTunes, sorry, and Google Play. Not to be confused with, we do have a reporting app on there that people have been trying to download, but that's for reporters. So if it says reporters, it's not for you. But yes, so if we are able to continue HabScope, it would be excellent to have a reporter at every beach that we do have beach conditions.
Yeah, and you can check with Mote's volunteer office. We know they had a link for people to just express their interest. If you're interested in Tracy's HabScope, Tracy's part of the HabScope project with all these partners. It's not just Tracy's, it's NOAA and NASA and GCU's and lots of great people. Oh, okay, so we got another question. Where are the deceased marine life found in the bloom areas taken to? Where do they go?
Well, first they come here. So they come to Mote, and we do our necropsy, postmortem exam, animal autopsy, and then most of them go to the landfill. If an animal's been humanely euthanized, or we've given that animal some sort of chemical which makes it unsafe to go there, then we have other means to dispose of it. But for the most part, everything's going to the landfill.
Mm-hmm. And I'm getting one question here about the cell of red tide, which is the Karenia brevis single-celled organism. People are asking if the release of water with nutrients and other chemical changes from Lake Okeechobee in particular, could it cause the cell to mutate? That's a good question.
That's a mutate. That probably not. Probably what would happen more likely, just that amount of fresh water coming out of Caloosahatchee, if that happened to land in an area where there was a lot of Karenia brevis in a bloom patch, probably lyse the cells.
It would break them open?
It'd break them open, which can release toxin, and that could cause a blast of toxin to occur. It'd have to be a fairly sudden and a fairly drastic change in salinity. But generally, so the salinity that we typically find in the Gulf of Mexico is somewhere in like 35, 36 parts per thousand. It can vary, of course, especially near inlets when you have freshwater flows. And Karenia can generally go down to about 25 parts per thousand. As an example, if you get into the upper parts of estuaries, you can get down as low as 15. The mid estuaries maybe 20 parts per thousand. So generally, Karenia would not really like that fresh water blast coming out of an inlet.
Another complication to all this stuff we have to figure out. Okay, do we know anything about red tide health effects for pregnant women? Have we heard anything to that effect? And Dr. Pierce or Tracy?
Yes. We had a scientist here who now is GCU's, is Dr. Barbara Kirkpatrick, and she is also a health scientist, working with a lot of the medical community, and it's found, they didn't do this research, found that the red tide toxin can be passed through the placenta to the baby, and so it's a good idea for pregnant women not to go to the beach if there's a lot of toxin.
Thank you. That's great information. So we have a question. Oh,
I'm sorry. The other side of that was, of course, the shellfish, which are the filter feeding organism, clams and oysters, accumulate high amounts of toxins because they filter out individual cells, so they become very toxic. So it's important not to collect shellfish during a red tide bloom and eat them. You get very sick, gastrointestinal as well as neurological effects. However, we have to say seafood restaurants and commercial areas are safe to eat seafood.
Yes. Now we have another question, a bioaccumulation related question just for you, Rich. Okay. We wanted to know if the animals that are affected by red tide are affected just by their straight up exposure to the algae in their environment, or whether it's the bioaccumulation in other animals that they eat that's a problem. Is it either, both, or what can you say about that?
Well, it's all of the above. It depends upon how the organism lives, how it breathes, and how it gets its food. Fish swimming through a red tide area, if their gills will lyse the cells. The cells go right into their bloodstream, and they are very neurologically affected. The manatees, for example, they eat seagrasses, and there are little critters that live on the seagrasses that filter and accumulate the toxins. So when the manatees eat the seagrasses, they're eating little creatures that are just full of neurotoxins, and so they get sick. Gretchen tells me that the dolphins get it from the fish that they eat. Now, the fish primarily don't have a lot of toxin in their flesh. Most of it is in their gut and their viscera. But we still don't recommend people to catch fish, even if they're alive in a red tide area. And then, of course, I'll let Gretchen talk to you about the sea turtles, because there's different species that eat different things.
Oh, yeah.
Yeah, so for the loggerhead and the Kemp's Ridley sea turtles, they're primarily crab eaters. We actually had a student a few years ago publish on her REU work where we saw that some of our younger Kemp's Ridley sea turtles during times of red tide were eating more fish. So we're thinking that might be a scavenging. You know, it's much easier to catch a slow dying or dead fish than it is a spunky crab. So, and then the green sea turtles that we see, they are they eat seagrass. Interestingly, they don't tend to show as high a levels of bryotoxin, and sometimes none at all, even though we're getting them in this area, but we do still think that it's somehow related. So.
Thank you. We had a question, someone was asking if we could please revisit the differences between Karenia brevis red tide and blue-green algae. For those who are just tuning in and weren't here at the very beginning, Vince, could you revisit that for us?
Sure, I'll try to be briefer this time.
Yeah.
So the blue-green algae, they're a type of cyanobacteria, photosynthetic cyanobacteria, or a type of bacteria, photosynthetic cyanobacteria.
They use the energy of the sun.
They use the energy of the sun, like the other phytoplankton, like Karenia brevis.
Like
they use
plants.
the energy, like plants, use the energy of the sun to create their own food source. And the ones that cause problems that we see in Lake Okeechobee and resulting from the outflows of Okeechobee and the Caloosahatchee and St. Lucie systems. Those are distinct. Those are freshwater, and they're distinct from our red tide Karenia brevis, which is a different group of phytoplankton altogether. It's a dinoflagellate. The cyanobacteria tend to occur in those blooms tend to be very multi-species. They can be many species, some of which are toxic, some of which are not. Some of the toxic species are sometimes toxic and sometimes not. Our Karenia brevis, even though it can vary, generally when we have a certain level of Karenia brevis, we expect to see toxic effects. You know, dead fish, respiratory irritation, and so forth. So that's the main difference. And Karenia brevis is a marine organism. As I said before about the salinity, it doesn't really like very low salinities.
Yeah, marine meaning living in the ocean, living in
Living in salt water.
Salt water likes that salinity. We had a question about when we have a deceased animal and we conduct a necropsy, which is an animal autopsy, and we take samples. What happens to the results? Do they go somewhere where people can find them, or can people ask us? I mean,
People can certainly ask. You know, we don't do our science in a bubble. The only way we can learn is by sharing it. The first places they go are our partners with Florida Fish and Wildlife and NOAA Fisheries. Those are the people that authorize us to do this work. You know, there's not a lot people can get from the gross necropsy reports, and again, it takes several weeks. And right now, the animals that at least the dolphins we've recovered, the only thing that we can really sample for to test is for that bryotoxin or the toxic mode. They're unfortunately too decomposed for us to get anything else.
So yeah, that makes it much harder if they're in poor condition. But you did mention gross necropsy-wise that you had seen fish in some of their bellies. So that's of interest because it's a possible route that they could get Karenia, right?
Right, and that is our most strong possibility right now, is that these are animals that were affected by red tide. They're not animals that live here in Sarasota Bay as part of our long-term resident community that's really well understood through Chicago Zoological Society's Sarasota Dolphin Research Program. They're one of the 9. They had one photo of its dorsal fin from 2016 near the Venice Fishing Pier. So we're thinking these are animals that are coming in from a little farther offshore. And again, that's also been pretty typical of what we've seen historically with these unusual mortality events that have affected dolphins in occurrence with HABs, harmful algal blooms. Sorry.
We have another question coming in here. We're checking with the studio. I think that's all the questions we have for now. Did anybody have anything they wanted to say before we wrap up? Last chance. You guys are great. Thank you for doing this. And thank you guys for all tuning in, for listening to us, for asking wonderful questions. You're all honorary scientists. Thank you for talking with us today. And you can email us any questions that we didn't answer today at info@mote.org. Again, that's info@mote.org. And you can follow us on Facebook and other social media. And we're also going to post this video to YouTube. So check out Mote's YouTube page, youtube.com/Mote Marine Lab, I believe it is. And you can find this and share it with your friends who are looking for information on red tide. And again, you can go to mote.org/red tide to find information and other sources of information from all the different partners we work with. Thanks again, everyone.